Hi, everybody. I'm Dr. Borsier. Um, I'm the one who's going to be on the other side of that screen this semester, and so I wanted to make a quick little video welcoming you to microbiology. So welcome to microbiology. I am Dr. Borsier. Technical difficulties. My computer is not behaving properly, so I'm trying to do this via Zoom, which is an unusual way of trying to do this. So I'm Dr. Boar Sear. I keep saying boar is in the pig, sear is in what you do with the pig. Boar, sear, boar, sear. I really just go by Dr. B though, because it's just so much easier. So who am I? I am born and raised in Michigan to a microbiologist. That's my parents there, microbiologist and an Austrian engineer. I grew up in the Thumb out in the country. I went to U of M Flint for undergrad. I went to Michigan State for graduate school for my doctorate degree. I currently live with my husband in Mason, which is south of Lansing. In fact, this is my Mason downstairs office that I am now using since I'm no longer in Grand Rapids. Usually during the school year, I live in a small condo in Grand Rapids that's right in Heritage Hill, which is within walking distance from campus. And so I do truly walk to school every day, uphill both ways in the snow. If you know Grand Rapids, it really is uphill both ways. I usually would be spending my summers geocaching, which is a fun hobby, and at home here in Mason, hanging out with my bees. Let's see, we're gonna put me, stick the talking head in the corner. Hanging out with my bees, and so in fact, when I am done working with you on any given day, I plan to be out in my apiary, which is just out that way. And I usually have between 2 and 6 hives at all times. So yeah, I guess I really am Dr. B, right? Although I have lots of critters all the time. I used to work with Australian Cattle Dog Rescue. I had 3 dogs of my own, but unfortunately they passed away at 11, 12, and 14 years old, and that was pretty tough, I have to say. They all went all at once, and the kitties did not enjoy condo living, so in October I adopted Pearl, which is my 11-year-old red heeler from Animal Control, and I'll tell you, she is fantastic. She's keeping me company most days, and usually right here by my side at the computer. So overview of our course, just really briefly, we have 750 points total for 5 units, and that's going to be good for you because our lab portion is 25% of your grade and our lecture portion is 75%. So if it's 750 points, how did that happen? Let me just fix it. 750 points. And each topic is going to have 2 or 3, or each unit is going to have 2 or 3 topics, and each topic will have a guide. And I do have a pretty lengthy explanation for the guides on Blackboard, but basically they're your reading guide, your note-taking guide, and your study guide. I don't ask you to turn them in, but they are the testable material for each of the topics and each of the units. And at the end of each topic, there will be a quiz. Quizzes are 25 points. I do automatically drop your 3 lowest quiz scores. It's already set up in Blackboard. And the quizzes will be over just that topic, but they're going to look like what's on the exam. So there'll be 25 points, true, false, matching, multiple choice, short answer, things like a fill in the blank. And then the exams are going to be over all the topics in that unit, and they're going to look very much the same. The quizzes and the exams are going to look the same, and that's intentional. There will also be homework assignments. Now the homework, it probably isn't going to look like homework that you might be expecting. I use homework as a tool for you to study, not as a, you find the right answer and then I check whether or not the answer was right kind of homework. These are more for you to take the information that you have learned and apply it and demonstrate that you understand by applying that information. Sometimes actually our homework is going to be used as a tool for studying before we've learned the information. So a way of studying and working through something in real time. There are also going to be participation points for posting on the boards. There are 3 required posts for 5 points each. Homework is about 10 points each. There are a few that are 5. Participation's 5 points each with the 3 required ones, and then there will be one that is, or I'm sorry, there will be 3 that are optional for extra credit. Next is, I just want to show you a picture here of our units, our overview of our semester. We're going to start with Unit 1, which is kind of scope and foundation. So scope and history of microbiology, what are we going to talk about, how did we get here, and then our foundations review are going to be the things that you really should have gotten in a previous biology course. That we're going to build on. And so I'm going to assume that when I say a polysaccharide, or I say translation to protein, you're going to know what those things are. And somewhere along the lines, you have probably learned this information. And so we're just going to kind of lay it out as this is the stuff that is our foundation. And so that's Unit 1. Unit 1 is actually going to be our week 1. It's going to be a pretty fast and short unit. And then we'll have an exam on our second week. Then we're going to move on to the kind of meat and potatoes of microbiology, right? The real hearty information. And we're going to start out with looking at the cell or unit structure. So prokaryotes and eukaryotes have a cell. And so we're going to look at the difference between us and them. We're eukaryotes. We're going to look at the difference between us and prokaryotes in terms of the cell structure and what some of those things mean. And then we're also going to look at life cycle, and we're also going to look at viruses. Viruses do not have a cell, and so we're going to call them a unit structure. So that's why this is a cell or unit structure, and we're going to look at viruses. It's very important for us to learn these 3 major cell or unit structures so that we can start to apply that information. So everything is going to build on our previous information. Everything is cumulative in some respect because it's continually building. Because next we're going to look at genotype to phenotype, why microbes grow where they do. So we're going to look at genetics. We're going to look at how microbes can acquire genes, manipulate their genes, share genes. And then we're going to look at some of those gene products. If you have a certain gene, then that means that you are going to metabolize in a certain way. And metabolism is going to be important for growth and nutrition. And so then we're going to look at our nutrition, our ecology, and growth patterns of different microbes. Once we can understand our cells and the way that they grow, then we can really start to apply it. And we're going to apply that in Unit 5. And now suddenly you're noticing, my goodness, Unit 5 comes between 3 and 4. That's intentional. That's because in my face-to-face lecture in Unit 4, which is immunology, we would actually be designing the vaccine and then presenting our designs. And so I wanted to have Unit 4 early enough so that we would have time throughout the semester for that. But in an online setting, first of all, the logistics of doing a group project would be absolutely crazy. And also, it actually works out quite nicely for us to build to this unit, which is immunology. So we're going to stick Unit 5 in the middle. That's going to be our control. So once we know about cell types and how they grow, then we can look at how we can control their growth. So we're going to look first at physical and chemical means of control. And then we're going to look at antimicrobial chemotherapy, which is drugs, antibiotics and antivirals and antihomintic drugs. And then we're going to get to our immunology. And our immunology, first we're going to actually look at good things about microbes. So you're going to learn in our very first lecture that most of our relationships with microbes are actually good. And so we're going to look at our good, healthy, resident flora that's on us and in us at all times and does good things for us. And then we're going to talk about how is it possible then for a microbe to cause an infection. And we're going to look at the process of infection and disease. We're going to look at the difference between true pathogens and opportunistic pathogens and the virulence factors that they might have. And then we're going to look at our defenses. We're going to look at the things that we have that protect us from those pathogens. And so we're going to look at our innate defenses, and then we're going to look at our adaptive and acquired defenses. And our adaptive and acquired defenses are the ones that give us memory. And memory is really good because that means that we won't be able to get the same infection twice. And so we're going to look at immunoglobulin replacement therapy or immunotherapy, which is a treatment based on this memory production. And then we're also going to look at vaccines. And so you'll notice here throughout the different units, there are these green boxes. Those are your homework assignments, and they're peppered in. Sometimes our homework is going to be to help serve the learning, and sometimes it's going to be to help apply the learning. So it goes either before or after the topic. And you'll also notice at the end here, after our last exam, we're going to have a case study, and we're going to do that case study homework across the entire semester. So we're going to take everything that we learned and looked at an animal bite and apply what we've learned towards analyzing this particular case. And then lastly, you're going to have this prokaryote information sheet resubmission. It's actually your first homework. Everybody's going to get assigned a microbe, and you're going to become an expert on that microbe. You're going to learn all about its cellular unit structure in the very first assignment. And then as we go throughout the semester, you're going to be taking the information that we learn in each topic and applying it to your specific microbe. So there's a series of questions that follow that initial assignment. And each time we go through a unit, there will be something to apply to your information sheet. And at the end then, you will have this all-encompassing information sheet about one microbe that you have now become an expert in, and then you'll resubmit that at the end of the semester. So that is our semester in a nutshell. As I mentioned, I am Dr. Borsier. I'm the face you'll be seeing. I hope to get to know each one of you soon, and I'm glad that we're together for this awesome microbiology course, and we'll see you soon. Bye-bye.